So biomarkers are molecules that you can detect in the blood, in the urine, or in tissue. And we can use them to try and diagnose patients or help to diagnose patients with prostate cancer. However, we know that not all biomarkers are fantastic. So you may have heard of prostate-specific antigen, or PSA, that we detect in the blood of patients. PSA is good, but it doesn't detect all patients with prostate cancer, and it's raised in the blood of some patients who don't have prostate cancer, causing them unnecessary worry and concern. So we know that genetically prostate cancer isn't caused by changes in just one single genetic component, and actually lots of different genes are altered when prostate cancer develops. So we don't just need one biomarker, we need lots of different biomarkers in order to improve the way that we diagnose prostate cancer. In the ReIMAGINE study, we're aiming to look at lots of combinations of markers and combine them with MRI in order to improve the way that we diagnose prostate cancer, and it's going to revolutionise the way that we do that.
So this is just an example of a prostate biopsy, and we have, tiny cores of tissue, and in the past we used, very basic technology, and, about 10 years ago, that's the only thing we need to know. Nowadays we are very interested in understand what happened between the tumor and, what makes a tumor more aggressive or less aggressive. At the end, we can also understand, just construct 3D dimensional models, 3 dimension, and we can see, which areas of the tumor they are going to be more aggressive, and we can do more genomics in those areas and treat those areas.
One way of looking at the ReIMAGINE consortium is that we're in the business of improving prediction. Predicting how a man's prostate cancer will behave if it's not treated, or indeed if it's subjected to specific treatments. And this will ultimately minimize under-treatment and over-treatment. Prediction is hard, but there are several examples where notable improvements have been made. And the factor that has allowed these major improvements is information. Large amounts of accurate data. My team will be present at the bedside to collect tissue biopsies, blood, and urine from all participants. These will be processed very carefully and rapidly to the highest of standards, and then frozen down and shared with several academic partners and commercial companies to extract the maximum amount of information from those samples. Then by integrating all that data about the mechanics of a man's tumor with all the other elements of information we extracted in the consortium, we will build much more accurate prediction models.